Hello students, myself Popere KS, Assistant Professor Botany at Raj Shikshan Sanstha's RBNB College, Sri Rampur. In this lecture, we are going to study about translation, that is mechanism of translation in case of prokaryote. And now what is translation? Translation, it is a process in which mRNA used as a template for the synthesis of the protein or a polypeptide. So in translation, this mRNA will be used as a template for the synthesis of the protein or a polypeptide chain. So this is protein. So for the synthesis of this protein, there are different things are required. These are the things which are required for protein synthesis are: first is mRNA. So mRNA, then ribosome, then tRNA. So these are the important things which are important for translation to occur. Now this ribosome, in case of prokaryote and eukaryote, this ribosome, they are made up of two different subunits, a smaller one and larger one. So in case of the prokaryote, it is so this ribosome unit, the small unit in case of prokaryote is 30 plus 50 is the large subunit, that makes 70S, and in case of eukaryote it is 80S, that is of 40S smaller and the 60S larger subunit. So this makes the ribosome, that is the two subunit make a ribosome. Now after this the tRNA. Now tRNA, this will bring the different amino acid for the synthesis of the protein. So this tRNA will carry the different amino acid during the protein synthesis process. Now we will now start with the actual translation process or the which occurs during this. So first is there is So first step will be the activation of the amino acid. Now for the activation of amino acid, there is requirement of ATP and one enzyme called as amino acid, that is amino acid synthetase. So for this, suppose this is an amino acid. So for this synthesis, that is activation of the amino acid, there is requirement of ATP and amino acid synthetase. So now the activated amino acid, it will be called as amino acid AMP. Now this is the activated amino acid. Now next step will be this, the loading of this activated amino acid to the, that is to the tRNA. So the attachment of this amino acid to the acceptor arm of the tRNA, and it takes place by an enzyme called as amino, that is the amino acid synthetase. So after this there will be loading of this amino acid to the tRNA by an enzyme amino acid synthetase, and the step is known as amino acylation of the tRNA. So next step is the So first is the activation of amino acid, and next then there will be the amino acylation of the tRNA, that is the loading of the activated amino acid to the tRNA, that is to the initiate tRNA. Now next we will see this amino acylation of the tRNA. Now this complex is called as amino acyl tRNA complex. Amino acid with tRNA, this complex is called as amino acyl tRNA complex. Now this complex is further important for the synthesis process. Now next we will see the initiation step. After this activation and loading, there will be next step, it is called as initiation. For this initiation step, there will be requirement of firstly the subunit of the ribosome. So ribosome, it is made up of, in case of the prokaryote, it is made up of two subunits, that is 30 and 50. So first there will be that is the assembly of 30S subunit. The ribosome 30 subunit will assemble. So what will happen? Firstly, suppose this is a ribosome. Now this will be a 30S subunit and this one is 50S subunit. Now this makes a 70S complex. This 70S complex of the ribosome, it has three sides, that is E, P and A side. Now it will be present at here, E, P and A side. So these are the three sides of the ribosome. Now they are important for the further protein synthesis process. Now next, what will be there? Firstly, there will be assembly of 30S subunit. So firstly there will be assembly of 30S subunit of the ribosome and mRNA and initiation factor 3. There are different initiation factors which are required for the protein during the initiation step of the translation. So firstly there will be assembly of now this is mRNA, this is the 30S subunit, and to this one initiation factor will be binding, that is initiation factor 3. Now this is the first complex which is forming. After that, now next will be, there is this complex is called as 30S So this is 30S mRNA IF3 complex. Now after this, simultaneously there will be one more assembly, that is of the initiator tRNA. So next assembly will be of the initiator tRNA containing an amino acid, that is the methionine. And one more initiation factor will be attached to this initiator tRNA, that is IF2. Now to this complex, the initiation factor which is binding is IF3, and now to this initiator tRNA initiation factor 2 is binding with one GTP. So GTP, it is attached to the initiator tRNA. So after this, now there will be formation. So after this there will be binding of this two complexes, that is the 30S mRNA complex and the complex of initiator tRNA with GTP and IF2. Now this is called as pre-initiation complex. So now these two complexes, the both has combined and they are forming a they have the presence of mRNA which is in 5' to 3' direction. Now this mRNA will bind on the ribosome at a specific site so that the codon AU, that is AUG, it is a start codon. Now this on mRNA AUG will be there and it will bind to the ribosome in such a way that this start codon will be placed in the, that is in the P site of the ribosome. So the first initiator tRNA which will be coming in the P site, that is, so this first initiator tRNA, it is present in this site, that is the P site containing a amino acid, that is the methionine. So on mRNA there will be AUG and it will form the codon, that is U, A and C. So the codon on this mRNA will be AUG and the anticodon on the this tRNA will be UAC. Now there will be codon anticodon interaction and in this way the protein will be after this interaction the amino acid will get incorporated into the polypeptide chain. Now after this, so this is the initiation stage. After that, next is the elongation. During the elongation there are two elongation factors, that is EFG, GTP. So these are the two elongation factors, that is EFTU, GTP, EFG, GTP. Now this will help in the elongation process of this protein synthesis. So what will happen? Firstly, now in the P site there is presence of initiator tRNA carrying the amino acid, that is methionine. Now what will happen? The next tRNA will come and it will come at a, that is A side. So next tRNA carrying the next amino acid, and this amino acid will be this amino acid will be on the basis of the codon which will be present on the mRNA, that is the next codon. On the basis of that, this amino acid will be coming along with the tRNA. So to this tRNA is bring by this factor, that is EFTU GTP. So the next tRNA, it is bring by this factor, that is EFTU GTP. So as the tRNA, the anticodon and codon interaction will take place in the A site. Now what will happen? This EF-TU GTP, it will convert into EF-TU GDP, and so after this, that is the GTP conversion into GDP, what will happen? This tRNA will be present now. Already in the P site there is presence of initiator tRNA, and now as the EF-TU GTP, it will converted into EF-TU GDP. What will happen? This tRNA will move this side. As this EF-TU GTP, it is leaving, what it will do? It will bind the tRNA in such a position that this tRNA's that is the head will be towards or in the P side, and its leg is in the A side. So how this tRNA will be there? That its head will be in the P side and leg is in the A side. So now what will happen? This EF-TU will release in the form of EF-TU GTP. Now it will remove from this A side. Now next what will happen? This next elongation factor, that is EF-T GTP, it comes and it will bind to this, that is to this the tRNA which will be present in the A side. It will bind here, and now what will happen? Here there is presence of the first amino acid, that is A1 or the methionine. Now there is presence of one more amino acid. Suppose we call it as A2. So what will happen? There is a peptide bond formation between these two amino acid, and now this EFG GTP, this elongation factor, now it has bind to this side, that is the A side, carrying the next tRNA. Now what will happen? This EF-TU GTP, it will get converted into EFG GDP. So after this will bind the tRNA when it is in the hybrid state. Hybrid state means half portion is present in the P side and half portion in the A side. Now after the conversion of GTP to GDP, what will happen? The mRNA, it is present in 5 to 3' direction, so it will move one codon. So this mRNA, the first side which is present in the P side moves to the E side. Now A side will move to the P side, and new codon will come into the A side. So this, while it is converted, that is EF-T GTP, it is converted from GTP to GDP. Now there is the movement of one codon from the mRNA, and this tRNA, that is the initiator tRNA, it will move into the A side, and the second tRNA, it will come into this side. Now this E side, it is also called as the exit side. Now the first initiator tRNA, which will come into the E side, it will release from this side, and the second tRNA, it will go to the P side. Likewise, now for the next round, there will be next tRNA carrying the next amino acid on the basis of the codon present on the mRNA. So this is how the elongation process will continue. So this is about the elongation. Now one more step is there, that is the termination. So last step is termination. Now for this termination process, there are different that is the signals due to which, or termination codon or stop codon, due to which what will happen? The protein synthesis will stop. So these are so these three are the stop codon. So when this codon is present on the mRNA, suppose these are the codon. If this 3 codon, these are called as a stop codon. If they are present on the mRNA, now at that time the protein synthesis will stop. So, and there are one more, these are stop codon. Now one more thing is there, that is release factor. Now this release factor are there are 3 release factor 1, 2 and 3. Now these are the factor which will recognize the stop codon, that is UAA, UAG and UGA. Now this release factor 1, it recognizes the stop codon UAA and UAG. Release factor 2, it recognizes the codon UAA and UGA, and this release factor 3, it can recognize any of the codon depending on the situation. So it can bind to any of the stop codon. So after the recognition, what will happen? This release factor will recognize the stop codon and it will terminate the protein synthesis process, and the ester bond which will be present on the, that is on the polypeptide chain of the protein, it will also be cleaved by this release factor. Due to this release factor, the ester bond which is present in the polypeptide, it will be broken down, and lastly the ribosomal subunit, that is the 2 subunit of the ribosome, they also dissociate, and now they are available for the next round of translation. So this is how the translation process or the mechanism of translation in case of the prokaryote. So firstly, it will start with the activation of the amino acid. Now the activated amino acid, it will be loaded on the acceptor arm of the tRNA. Now after that, the initiation process will occur, during which the different, that is the 30S subunit, it will bind to the mRNA, and initiation factor 2, it will bind. After that, then there is a next step, that is the elongation. There are different elongation factor, that is elongation factor EF-TU-GTP, EF-G-GTP. Now they will come and bind at a specific position, due to which there is anticodon-codon interaction, and the mRNA will move, that is one codon, and this is how the elongation process will take place. After then there is termination process. During that, there are different stop codon when they are present on the mRNA, and when the ribosome reaches the stop codon, at that time the ribosome subunit, due to the presence of the release factor, they dissociate, and the termination process of the protein synthesis stop there only. So this is how the translation takes place in case of the prokaryote. Thank you.